Conclusion: the return to structure. Biological function does not originate from isolated pathways or individual molecules acting in abstraction. It emerges from the physical organization of the system, where membranes define boundaries, control exchange, and establish the conditions under which all processes operate. When this structure is stable, signaling is precise, transport is controlled, and energy production proceeds without loss. When it degrades, the same processes persist but produce inconsistent and often conflicting outputs. The difference is not in the presence of activity, but in the conditions that shape how that activity unfolds. The collapse into calorie-based thinking removed structure from the model and replaced it with accounting. Energy intake and expenditure became the focus, while the architecture that determines how energy is used was ignored. This shift explains why interventions built on energy balance alone fail to produce consistent outcomes. Without stable membranes, inputs are processed under variable conditions, leading to unpredictable behavior. Restoring structure returns the model to the level where outcomes are determined, allowing the system to operate under defined constraints. Membranes function as the governing layer that integrates all systems. They regulate permeability, maintain gradients, and provide the environment in which receptors, transporters, and enzymes operate. Lipid composition determines how these functions behave, influencing mechanical stability, electrical properties, and signaling dynamics. This places lipids at the center of biological organization, not as passive energy sources, but as structural components that define system behavior over time. The body maintains itself through continuous turnover, replacing lipid components based on available substrates and environmental conditions. This process allows adaptation, but also introduces vulnerability. Without control over inputs, remodeling cycles incorporate instability into the structure, gradually altering function. With consistent input of stable substrates and reduced disruption, the same cycles reinforce integrity, aligning membranes across tissues and restoring coordinated behavior. System-wide coordination depends on shared structural conditions. Metabolic, immune, and neural systems communicate through signals that require consistent interpretation. When membranes differ, these systems operate under conflicting rules, producing fragmented outputs. As membrane properties converge, signaling aligns, energy production stabilizes, and feedback mechanisms regain accuracy. The body shifts from a state of compensation to one of coordination, where processes reinforce rather than interfere with one another. Long-term maintenance ensures that this state persists. Continuous control of substrate quality, oxidative load, and energy availability allows remodeling cycles to preserve structure rather than erode it. Environmental pressures remain, but the system adapts to them without losing stability because the underlying architecture is maintained. This prevents regression and supports consistent function across varying conditions. The return to structure reframes biology at its foundation. It moves the focus from inputs measured in isolation to the physical system that determines how those inputs are processed. By maintaining membrane integrity and controlling the conditions that shape remodeling, the body operates with predictable, efficient, and coordinated behavior. Outcomes follow from structure, and when structure is restored, the system returns to a state where its functions align with the demands placed upon it. Biology as a material system. Biology operates through physical materials organized into functional structures, not through abstract pathways detached from their environment. Cells are constructed from lipids, proteins, and aqueous compartments arranged in a way that constrains how molecules move, interact, and transform. Every outcome in the body reflects how these materials are assembled and how they behave under given conditions. Signals, reactions, and energy flow all depend on this arrangement. Without the correct material properties, the same inputs produce different outputs because the system itself has changed. Membranes define the primary material boundary of the system. They separate internal conditions from the external environment and regulate what crosses that boundary. The lipid composition of these membranes determines permeability, mechanical strength, and electrical behavior. A tightly packed membrane limits uncontrolled movement, preserving gradients that drive transport and energy production. A disordered membrane allows leakage, dissipating gradients and altering how signals are interpreted. This boundary is not passive. It is the interface that shapes all internal activity. Proteins operate within this material framework and depend on it for proper function. Receptors require specific lipid environments to maintain their conformation and to cluster with associated signaling components. Transporters depend on membrane thickness and fluidity to cycle between states that move substrates across the bilayer. Enzymes embedded in membranes interact with lipids that influence their activity and stability. When the surrounding material changes, protein behavior changes with it, even if the protein itself remains structurally intact. Energy production is constrained by material properties. Mitochondrial membranes must contain proton gradients to drive ADP synthesis. This containment depends on lipid composition, particularly the presence of cardiolipin and tightly packed phospholipids that reduce leakage. When the material fails to maintain this barrier, energy dissipates as heat rather than being captured as ADP. The efficiency of energy conversion therefore reflects the integrity of the membrane rather than the availability of substrates alone. Signaling systems rely on spatial organization within membranes. Lipid microdomains create regions where specific proteins concentrate, allowing signals to be initiated and propagated efficiently. These domains depend on lipid interactions that define their size, stability, and composition. When the material is consistent, signaling pathways operate with defined timing and strength. When is not, signals become diffuse or exaggerated, leading to variability in response. Transport processes are governed by gradients that exist because of material boundaries. Ion concentrations, substrate levels, and electrical potential differences are maintained by limiting uncontrolled exchange. Membrane composition determines how well these gradients are preserved. Stable materials support controlled transport through channels and carriers, while unstable materials allow passive leakage that disrupts distribution and forces compensatory activity. Adaptation occurs through changes in material composition. Cells adjust lipid content, protein expression, and structural organization in response to environmental conditions. These changes are incorporated through continuous turnover, meaning that the material state of the system reflects cumulative inputs over time. Adaptation therefore modifies structure first, and function follows as a consequence of that structural shift. Damage is also material in nature. Oxidation, mechanical stress, and chemical interactions alter the physical properties of lipids and proteins, disrupting their organization. Repair involves removing damaged components and replacing them with new materials that restore the original structure. The success of this process depends on both the availability of appropriate substrates and the energy required to perform the replacement. Viewing biology as a material system places emphasis on what the body is made of and how those materials are arranged. It explains why identical biochemical pathways can produce different outcomes under different structural conditions and why long-term inputs shape function by altering composition. When the material properties of the system are stable, processes operate within defined limits and produce consistent results. When they are not, variability emerges from the structure itself, influencing every level of function. Structure as the driver of all outcomes. Outcomes in biology follow from structure because every process is constrained by the physical conditions in which it occurs. Membranes define those conditions by setting permeability, organizing proteins, and maintaining gradients that enable transport and energy production. When this structure is consistent, inputs are processed within defined limits and produce predictable results. When it varies, the same inputs lead to different outputs because the underlying environment has changed. Function does not drift on its own. It follows the state of the material that supports it. Signaling behavior reflects membrane organization. Receptors activate, cluster, and terminate based on the local lipid environment that controls their mobility and interaction with downstream components. When membranes maintain stable packing and domain structure, signals are interpreted with consistent thresholds and timing. Variability in composition alters these parameters, shifting activation sensitivity and prolonging or shortening responses. The signal itself remains unchanged, but its effect diverges because the structure that receives it has shifted. Transport depends on structural containment. Gradients of ions and substrates exist only when membranes restrict uncontrolled movement. These gradients drive the entry and exit of materials through regulated pathways, allowing cells to maintain internal conditions distinct from their surroundings. When membrane integrity declines, leakage increases and gradients weaken, forcing compensatory mechanisms to restore balance. This alters distribution and increases energy demand, changing how substrates are delivered and used across the system. Energy production is determined by the ability of mitochondrial membranes to contain and utilize proton gradients. The electron transport chain generates a difference in charge and concentration that must be preserved to produce ADP. Structural defects in the membrane allow protons to return without driving ADP synthesis, reducing efficiency and increasing heat loss. Energy availability, therefore, depends on the physical integrity of the membrane rather than on substrate supply alone. Immune activity is shaped by structural recognition and signaling. Membranes present molecular patterns that determine how immune cells identify targets and initiate responses. Lipid mediators derived from membrane components control the intensity and duration of these responses. When structure is stable, recognition and resolution proceed in sequence. When it is not, signaling becomes prolonged or misdirected, leading to incomplete resolution or inappropriate activation. Hormonal responses depend on receptor environments and transport conditions. Hormones circulate with defined concentration and timing, but their effects are determined by how receptors respond within each tissue. Membrane composition sets the conditions for this response, influencing sensitivity and signal propagation. Structural consistency allows hormonal signals to produce coordinated effects across tissues, while variability creates divergence in response. Neural function relies on controlled electrical properties of membranes. Ion gradients and channel behavior determine how signals are generated and transmitted. Stable membrane structure preserves these gradients and supports consistent conduction. Structural disruption alters electrical behavior, introducing variability in signal transmission and timing. All systems converge on the same dependency. The body operates as a network of processes that require stable boundaries, controlled exchange, and consistent signaling environments. These requirements are met through membrane structure, which integrates lipid composition, protein organization, and energy dynamics into a single framework. Changes in this framework propagate through every level of function, altering outcomes without changing the inputs themselves. Structure therefore acts as the governing layer of biology. It determines how signals are interpreted, how substrates are distributed, and how energy is produced and used. By maintaining membrane integrity and controlling the conditions that shape it, the system operates within predictable limits. When structure degrades, variability emerges, and outcomes become inconsistent. The direction of change is set not by the processes themselves, but by the material state in which those processes occur. Lipids as the foundation of control. Control in biological systems does not originate from signaling molecules alone. It emerges from the material that defines how those signals are received, transmitted, and terminated. Lipids form that material. They construct the membranes that determine permeability, organize proteins into functional arrangements, and establish the electrical and mechanical conditions required for coordinated activity. Without stable lipid structure, control is lost even when signaling inputs remain unchanged because the system no longer interprets those inputs in a consistent way. Membrane permeability is the first layer of control. Lipid packing determines how easily ions and molecules cross the bilayer outside of regulated pathways. Tightly packed membranes restrict passive movement, preserving gradients that drive transport and electrical activity. When packing loosens, leakage increases, gradients weaken, and control shifts from regulated transporters to uncontrolled diffusion. This changes how substrates are distributed and how signals propagate, introducing variability that the system must compensate for through increased energy expenditure. Spatial organization of proteins depends on lipid composition. Receptors, channels, and enzymes are not randomly distributed. They localize within specific lipid environments that support their function. Cholesterol and sphingolipids create ordered regions where signaling complexes assemble, allowing rapid and coordinated activation. The surrounding lipid matrix determines how these domains form, how stable they remain, and how quickly they disperse after signaling is complete. Changes in lipid composition alter this organization, affecting both the strength and timing of signal transmission. Electrical control is also lipid-dependent. Membrane potential arises from a separation of charge across the bilayer, maintained by ion gradients and selective permeability. Lipids influence both the ability to sustain these gradients and the behavior of embedded ion channels. Stable membranes support consistent electrical properties, allowing predictable signal generation and conduction. Structural disruption alters ion leakage and channel function, changing how electrical signals are initiated and propagated. Energy production is governed by lipid structure within mitochondria. The inner mitochondrial membrane must contain proton gradients to convert electron flow into ATP. Cardiolipin and tightly packed phospholipids create the conditions required for this containment. When lipid composition shifts, proton leakage increases, reducing the efficiency of energy conversion. This affects every process that depends on ADP, linking energy availability directly to membrane integrity. Lipid-derived molecules extend control into signaling pathways. Fatty acids released from membranes serve as precursors for mediators that regulate inflammation, vascular tone, and immune activity. The types of lipids present determine which mediators are produced and how long they persist. This ties signaling outcomes to the structural composition of membranes, making lipid balance a determinant of system behavior rather than a passive background factor. Adaptation and regulation occur through continuous lipid turnover. Remodeling cycles replace existing fatty acids with those available in the cellular pool, gradually altering membrane composition. This process allows the system to adjust to environmental conditions, but it also means that long-term inputs shape control mechanisms over time. Stable inputs reinforce consistent control, while unstable inputs introduce variability that propagates through signaling, transport, and energy systems. Feedback mechanisms depend on lipid-defined conditions to operate correctly. Signals generated in response to changes in system state must be interpreted consistently across tissues to maintain balance. When lipid composition diverges, feedback becomes inaccurate, leading to overcorrection or persistent imbalance. Maintaining lipid integrity ensures that these regulatory loops function within expected parameters. Lipids therefore provide the foundation of control by defining the environment in which all biological processes occur. They regulate what enters and leaves cells, organize the components that transmit signals, and maintain the conditions required for energy production. By shaping these core functions, lipids determine how the system behaves as a whole. Control is not imposed on the system from above. It is built into the structure of the membranes that govern every interaction within it. Stability as the basis of health. Health reflects the ability of the system to produce consistent outputs under changing conditions. This consistency depends on structural stability, where membranes maintain controlled permeability, organized signaling environments, and contained energy gradients despite fluctuations in temperature, nutrient availability, and external stress. When structure is stable, processes operate within defined limits and return to baseline after perturbation. When it is not, variability accumulates, and the system shifts into continuous correction rather than controlled function. Membrane stability preserves gradients that define internal conditions. Ion concentrations, electrical potential, and substrate distribution all depend on the ability to restrict uncontrolled exchange. Stable lipid packing maintains these gradients, allowing transport systems to operate predictably. When membranes lose integrity, leakage increases, gradients weaken, and cells must expend additional energy to restore balance. This alters distribution patterns and reduces efficiency, affecting every downstream process that depends on controlled internal conditions. Signaling stability emerges from consistent receptor environments. Activation thresholds, clustering behavior, and termination kinetics depend on membrane composition. When these properties remain within a defined range, signals are interpreted with precision, producing proportional responses. Structural variability shifts these parameters, leading to exaggerated or diminished signaling that disrupts coordination between tissues. Stability ensures that signals maintain their meaning as they move through the system. Energy stability is determined by mitochondrial membrane integrity. ADP production requires containment of proton gradients and efficient electron transport. Stable membranes support this process, allowing energy output to match demand without excessive loss. Structural disruption increases proton leakage and reactive species generation, reducing efficiency and creating variability in energy availability. Consistent energy supply supports all maintenance, repair, and functional processes. Temporal stability allows processes to occur within defined windows. Activation and termination of signaling, repair cycles, and metabolic transitions depend on predictable timing. Stable membranes enable these processes to begin and end without distortion, preserving sequence and coordination. When timing varies between tissues, processes overlap or diverge, leading to interference and inefficiency. Resistance to stress is a consequence of structural stability. Mechanical forces, oxidative exposure, and metabolic demand all challenge membrane integrity. When membranes resist deformation and damage, the system can absorb these stresses without altering core function. This reduces the need for compensatory responses and allows rapid return to baseline conditions once stress is removed. System-wide coordination depends on shared stability across tissues. When membranes in different organs maintain similar properties, signals produce aligned effects and feedback mechanisms operate accurately. This allows metabolic, immune, and neural systems to function as an integrated network. Variability in structure disrupts this alignment, creating fragmentation where systems operate under different constraints. Long-term stability is maintained through continuous turnover that replaces degraded components with stable ones. Each remodeling cycle either reinforces or erodes structural integrity depending on substrate availability and environmental conditions. Sustained input of stable lipids and controlled oxidative load allow these cycles to converge toward consistent membrane composition, preserving function over time. Health, therefore, arises from the maintenance of stable structural conditions that govern all biological processes. By preserving membrane integrity, supporting energy production, and ensuring consistent signaling environments, the system operates with predictable and coordinated behavior. Stability allows the body to adapt to variation without losing control, making it the underlying basis from which reliable function emerges.